TNF on microglia density and morphology in the CA1 region of the hippocampus. Take it away.
Hello everyone, so as previously said, I'll be talking about the impact of TNF on microglia density and morphology in the CA1 region of the hippocampus. Now, to give some context to what all of this means, I need to start by talking about cognitive decline. So cognitive decline is what we characterize as unhealthy aging. It is a general decrease in mental faculties that is said to begin around the age of 60. This includes many different faculties, from memory to spatial reasoning, and all of these are things that somebody needs to be able to do well in order to live an independent and high-quality life as they get older. Now, it's a global issue. An estimated around 20% of older adults are going to be touched by some form of cognitive decline as we get older, and this is important because as it's a reality for all of humanity, everyone is going to be old at some point, and so everyone could eventually develop some kind of cognitive decline, which is why it's really important for us to understand what is the root cause of this illness and what can we do to alleviate it. So when we consider aging as a whole, it's actually a very complex problem with many different metabolic and cellular processes. So we want to focus on one aspect, which is chronic inflammation. And it's an accumulation of low-grade inflammatory signals in your blood that can dysregulate cells. So to give and link back to cognitive decline, one of these pro-inflammatory cytokines that we look at is tumor necrosis factor 1, or TNF. It's a cytokine that is involved in many cellular processes, from apoptosis to cell proliferation, and previous studies have found that as we get older, there is a higher amount of TNF circulating in our blood, and this higher amount of TNF is directly correlated to a higher prevalence of cognitive decline. So if you have more TNF, you have a higher chance of also developing cognitive decline. So in our own lab, we wanted to examine this in TNF knockout mice. So these are mice that do not have a global level of TNF, so there is no TNF circulating in their blood, but they do still have the receptors. So if you were to inject the mice, for example, with TNF for whatever reason, they could still react to it, but they do not produce it on their own, which gives us a good example of a species that won't have this chronic inflammation that we associate with cognitive decline. So to test their memory, we use something called the novel object recognition test. How this works is we put the mouse in a box with two identical objects, and we give it time to familiarize itself with the objects before putting it in a new box with one of the older objects, but also a new object that it hasn't had the chance to familiarize itself with yet. And we want to see which object is the mouse going to prefer. If its memory is more intact, it's obviously going to have a preference for the object that it hasn't had the chance to explore yet. If its memory is less intact, if there are symptoms of cognitive decline, then it might not show a preference for either one of them, or might even prefer the old one. So we used really old mice. By really old, we mean 20 to 22 months of age, which is very old for mice. And when you look at this graph here, the positive numbers indicate a preference for the novel object, so the new object that it hasn't had a chance to explore, and the negative numbers indicate a preference for the old object. So our TNF knockout mice, so our mice without chronic inflammation, showed a preference for the novel object, demonstrating that their memory was more intact than our wild-type mice that have normal levels of inflammation. So TNF probably has some kind of impact on cognition, a negative impact. The question now is, when we look at the brain, what is the source of TNF in the brain? And the answer to that is microglia. So, what are microglia? They are the resident macrophages of your central nervous system. They are essentially one of the main immune cells in your brain, and they have been there since development. So even early on, when we think back to neurogenesis, microglia are deeply involved in the process of allowing new neurons to colonize your parenchyma. They are frequently involved with crosstalk with other glial cells in your brain, so they are super involved. They're always getting the environment. And there's many changes that can happen to them as we get older, namely morphological changes. So one of the things that we observe is when we look at morphology of microglia, we look at two things. We look at their cell body, so the center part here, and we look at their arborization. They really need these processes to be very fluid and dynamic, so they can properly screen the environment for damage signals, for just cross-linking with other glial cells, making sure that everything is running as expected. As you get older, they have two distinct changes. Their cell bodies grow in size and their arborization reduces, which also impairs their function to do their job properly as these surveillors of your brain. There's another change that happens. When you are considered cognitive decline, there are certain regions of the brain that are more susceptible to it than others. One of these regions is the CA1 region of the hippocampus. It looks something like this. And it's linked to memory and spatial reasoning, which, as I mentioned before, are faculties that will decrease when you have cognitive decline. So when we look at microglia numbers in these regions of the brain, we actually find that there is an increase in density of microglia in these areas, which leads us to our hypothesis for this project. We think that if TNF is responsible for some of the changes linked to cognitive decline, microglia from TNF knockout mice will have lower microglia density and smaller somas and larger arborizations compared to wild-type mice, essentially matching more what you see in younger mice versus older mice. So, how do we test this? We use TNF knockout mice and wild-type mice, again, very old, 20 to 22 months, and we remove their brain, cut it, and we fixed it with two primary antibodies. So this is IBA1, which is ionized calcium binding adaptive molecule 1. And this molecule is a macrophage marker. It essentially helps us visualize the microglia as a whole. And then we also have TNF119, which is a microglia marker. It'll help differentiate microglia from other macrophages. So we added our secondary antibodies to actually visualize them. So when you see green, that's IBA1, our macrophage marker. When you see red, that's TNF119, our microglia marker. And we added DAPI, which is just a nucleus marker, helps us identify other cells that are not microglia in the area and look at different brain structures. And finally, we image them. So this is one of our example of our 60 magnification images. And there were two analyses that we wanted to do with these images. First is a density analysis, the second is a morphology analysis. I'm going to go over density first. How it works is we have our 20 magnification image right here, and we would use different references images to trace out the region of interest as such. And then we had something else to consider. I might have mentioned before that IBA1 is a marker for macrophages and microglia. So thanks to this, we can actually look at the presence of infiltrating macrophages in the brain. These are macrophages that come from the periphery or the blood around the brain that get called in when there's damage or when there's some kind of pathogen and infiltrate the brain's tissue. Now, they also have an increased presence with age. So we can see this is a second marker to see if there's any age-related changes that TNF is responsible for. So once we know all this, and once we've traced out the region, you can start counting each and every single individual cell to get a measure of density. So when you look at your different channels, you can have a microglia, and if you were to turn off the IBA1 channel, you can still see the red, which tells you this is a microglia, not a macrophage. Vice versa, with my infiltrating macrophage, when I turn off my IBA1 channel, I don't see the same red, which helps me differentiate the cells and allows me to count them more efficiently. So just to recap, because this is a lot, you trace the area, and you place dots on macrophages and microglia, and then there's two density measures. The first is amount of microglia per millimeter squared, and the second is what we call nearest neighbor distance. It is essentially just a measure of the average distance of one microglia to another. So if there's lower density, they should be more spaced apart. If there's higher density, they'll be closer together. And this is what we found. So old TNF knockout mice have lower microglia density compared to old wild-type mice. This confirms our hypothesis. So TNF does have some kind of impact on microglia density. We see the same thing with our NND values. Microglia from old TNF-KO mice have higher NND values, so their microglia are more spaced out than wild-type mice, again confirming they have some lower density. However, there is no difference in percent infiltration, so whatever is creating an increase in infiltrating macrophages, it is not because of TNF. Then we looked at morphology. So in order to do that, we traced out the area of the soma, so the cell body, and we traced out the arborization. And it also gave us an ability to compare their size by doing one over the other. And this is what we found. There is no significant difference in morphology between old TNF, KO, and wild-type mice. Now, we know that TNF can cause morphological changes when there's damage and when there's pathogens. They can swap microglia from a resting state to an activated state, which is a rounder morphology that we associate with aging. However, this requires a large influx of TNF and other pro-inflammatory markers. So with age, this change in morphology might actually be due not to an increase in activated microglia, but an increase in what we call dystrophic microglia. These are microglia that have taken up mucofuscin deposits from myelin, and myelin is this fat tube essentially that goes around your neurons to help increase conductivity of action potential. And so when microglia take up too much of this fat deposit, they tend to become dysfunctional. So this might explain the age-related morphology changes we see, not necessarily chronic inflammation, which is a strong conclusion. So TNF is linked to a change in microglia density, but not a change in infiltration or microglia morphology, which tells us that chronic inflammation is responsible for some, but not all of the changes that we see in the hippocampus with age. And of course, for a complete analysis of all of this, we also need to have younger mice. So this is something that our lab is currently working on. This will help us confirm, first of all, the age-related changes that other studies have seen, and also to see how early on does this chronic inflammation actually have an impact on our microglia. Next, microglia and morphology and density are not the full picture when it comes to microglia changes with ages. We also know that they show an increased production of pro-inflammatory cytokines as you get older. In order to test for this, we've been working on using a Microglia kit, that's a magnetic separation kit, to isolate microglia from whole brains in the hope of stimulating them with TNF, because as I mentioned before, our TNF knockout mice can still react to TNF, so if you stimulate with them TNF, they'll still be able to produce cytokines in response to this. So we're intending to use that to measure IL-6 levels, which is another prominent pro-inflammatory marker in chronic inflammation, with Alizee in hopes of getting a better picture of what the changes are with age. So I'd like to thank everybody in the lab for being super supportive and super welcoming this year, and that's the end of my presentation.
Alizee, anybody have any questions?
Yeah, it's a great talk. I want to ask, the behavioral assessment that you were talking about at the beginning, are the TNF knockouts, like how similar is that to like a younger mouse? Like is the knockout, I'm basically asking like how good is the knockout at like keeping them really from aging, right?
I haven't actually ever seen them, but from what other people in the lab have described, there is actually a pretty visually striking difference between our wild type and our TNF knockout mice when you look at them. Our TNF knockout mice, they have glossier fur, they just look healthier. Like chronic inflammation has more than just an impact on cognition, it also dysregulates everything in the body. So generally speaking, they're just closer to younger mice than these older wild type mice.
I might have missed this, but did you do this in both male and female mice? And do you expect differences in microglia morphology?
I did do it in male and female mice. I didn't present my results because we don't have that many wild type mice to compare them to, but we do expect to see some differences in morphology. I'm trying to remember, I'm pretty sure male mice usually have more microglia than female mice, or the other way around, but yes, this is something that our lab is definitely expecting to look into, the sex differences between the two groups.
I have a quick question. Is there any phenotype to these TNF knockout mice, like normally? Is there any apparent defects or